I have the pleasure of introducing our next speaker, Dr. Tcheandjieu. She is currently a postdoc at the Department of Medicine at Stanford School of Medicine and at the Palo Alto Epidemiology Research and Information Center for Genomics, where she studies the genetic risk factors for cardiovascular disease.
Thank you so much, Sarah and Dan, for inviting me to this amazing symposium. So today, I will talk about the benefit of diverse population for genetic discovery in coronary artery disease. CAD is a group of disease characterized by progressive plaque formation in the vessel wall. This process, called atherosclerosis, will result to a blood flow reduction in the artery, causing the heart to beat harder and faster. Myocardial infarction or heart attack is typically caused when a blood clot is incited by a rupture in the surface of the plaque. This process deprives the heart muscle downstream of the blood clot, leading to permanent muscle damage. CAD is the leading cause of death due to cardiovascular disease in the US and worldwide. The lifetime risk factor is higher in men than women. Although the prevalence of CAD is lower in non-Hispanic Black than in white, and you can see here, death due to CAD is disproportionately higher in non-Hispanic Blacks than any other ancestry group, as you can see in this picture. There are multiple risk factors for CAD, including genetic. However, genetic risks for CAD are not well understood. Up to date, genome-wide association studies have identified over 210 loci associated with CAD, mostly in non-Hispanic white, South Asian, and East Asian. No association has been reported in Black and Hispanic, mainly due to that lack of study or study with underpowered sample. This highlights the need of doing more research in genetic for CAD in this population. So today I will talk about a multi-ethnic GWAS and meta-analysis of CAD that include the Million Veteran Program and multiple external cohort. I will further extend my talk in understanding genetic variation, driving the difference in association for CAD across ancestry at the locus 9p21. 9p21 being the locus with the strongest association ever reported for CAD in white and other ancestry group. This slide provides an overview of the Million Veteran Program. The MVP is a nationwide multi-ethnic cohort of veterans in the US. Up to date, the cohort includes over 800,000 participants, among which we have genetic information for over 650,000 participants. However, the current work is based on about 408,000 participants for which we had genetic information at the time of analysis. Participant information include, among all, lifestyle survey and electronic health record that was able to be extended for over 10 years prior to the enrollment. The MVP have a particularity of being multi-ethnic. So in this context, ancestry assignment in participant was defined using HAIR, an algorithm that combines self-reported and genetic, self-reported ethnicity and genetic ancestry. In the context of this work, we include non-Hispanic white, non-Hispanic Black, and Hispanic. And our CAD phenotype was defined using ICD code, hospital discharge for acute myocardial infarction, or procedural code for revascularization. So I'll now talk about our multi-ethnic GWAS and meta-analysis that include MVP as a discovery cohort and multiple external cohort, as well as currently available GWAS for CAD. This flow chart represents our overall analysis. So we start off with GWAS within each ancestry group in the MVP as a discovery cohort. We then perform a meta-analysis in white only, which include MVP white, the UK Biobank, and cardiogram proceeded for the. These three data combined give us about 190,000 cases and over 585,000 control. We further extend to a trans-ethnic meta-analysis, which include all MVP ancestry group, UK Biobank, cardiogram proceeded, as well as the Biobank Japan. All these together brought up to over 250,000 cases and over 850,000 control. We finally extend our scope of discovery discovery for Blacks and Hispanic by selecting a subset of SNP for which we sought for replication in external cohort and combined the result from the outside cohort and MVP to perform a second stage meta-analysis. So in Black, MVP plus external cohort together gave us about over 26,000 cases and 100,000 control. In Hispanic, we have over 12,000 cases and 53,000 control. Our extended data set for these two ancestry groups include Emerge, WHI, Page, BioView, Penn Biobank, and other cohort. So this sequence plot represents our overall result of a genome-wide association analysis. From the outside of this plot to the inside, we have the transethnic meta-analysis. In blue, the meta-analysis in white. In Black, the meta-analysis in Hispanic in green, as well as the analysis in Black in red. So for our white-only meta-analysis, we identified 37 new CAD loci, including 4 loci in the X chromosome. And this is the first time CAD loci have been identified on the X chromosome. Among our new loci, we have TGF1P3, which was previously reported for lipid and is located on the X chromosome. And we also have a loci in FTO, which is a well-known obesity-related. For our transethnic meta-analysis, we've identified 17 new CAD loci, including 5 additional loci on the X chromosome. Among our discovery, we have a loci in PNPLA3, which is a well-known NAFLD loci, as well as TCF7L2, a well-known type 2 diabetes loci. So what we see in our transethnic meta-analysis is that by increasing the overall number of cases with 2,000 extra cases from this multi-ethnic group, we've leveraged 2 times more discovery than when we consider white-only meta-analysis. Our analysis in Black helped us identify 5 CAD loci, which are in LPL, LPA, FDG5, ApoE, and COR4A1. All these loci were previously reported in white. For Hispanic, we've identified 3 CAD loci, which are in FACTIA1, ApoE5, and ADAMTS7, again, previously reported in white. So in overall, we've identified 8 CAD loci in Black and Hispanic, all previously reported in white, as you can see with each of those arrows. But surprisingly, 9p21, which is so far the strongest association that ever reported for CAD in white, have not been replicated either in Hispanic and in Black, despite the pretty substantial number of cases that we have in each of those ancestry groups. So the question now is, why is that despite our case number, we are not able to get 9p21 reach genome-wide significance? So the next part of my talk will be on focusing on fine mapping of the locus 9p21 in a million veteran program. So 9p21 is the first locus to reach genome-wide significance for CAD. The locus was first reported in 2007 on one of the study that reported the locus was Wellcome Trust Case Consortium, which include over 2,000 cases. The locus is located in the short arm of a chromosome 9 and span about 60 kb. It contains CDKN2A and CDKN2B as well as RNIL, but up to date, it's still unclear what is the biological mechanism driving the association with CAD at the locus, and it's also unclear what is the causal gene or causal variant in this locus. Nevertheless, the association with SNP at 9p21 has been replicated at the genome-wide level in multiple other studies in white, in East Asian, in South Asian, but has not been reported at the GY level in Hispanic or Black. Mainly, there hasn't been a GY study for Hispanic, and the GY study in Black has been so far underpowered. So to understand why the association at 9p21 is not replicated in Black, we first hypothesize that maybe SNP in the locus might have a low allele frequency, and therefore, despite our large number of cases, we are still not powerful enough to be able to find any association. So here, this table represents a subset of SNP that has reached genome-wide significance in white over time and also in Asian population. And you can see here the allele frequency of those SNP in the combined MVP Black and our external cohort. And we see that the allele frequency are fairly common, but yet we are not able to reach genome-wide significance, even though for some of the SNP we are trending. So if we do a power calculation, assuming an odds ratio of 1.15, which is the average odds ratio reported in white, the prevalence of the disease as what we see in Black, and a genome-wide significance type 1 error with our case control ratio, what we see is that we already have over 90% power to identify a genome-wide significant association with SNP of allele frequency of 5% and above. So now the next question is this lack of association due to some form of heterogeneity that is caused by admixture in Black in MVP at this locus? So to answer this question, we perform a local ancestry inference at the locus 9p21 in Black and in Hispanic, and then we stratify our subgroup based on a proportion of ancestry and perform association testing at the SNP level, as well as perform some haplotype analysis to understand if there is some specific haplotype pattern that could explain this lack of association. So really briefly, local ancestry estimate consists of assigning to each individual the best ancestry at the chromosomal segment based on the selected reference population. So basically what we're trying to do is that as we walk over the chromosome, we are assigning to our target individual the ancestry for which the genetic pattern is similar to the genetic pattern that we see in our target population. So for this, for Black, we use British GBR as European from 1000 gene as reference variant, and European as African population from 1000 gene as a reference panel. And for Hispanic, additionally, we use Peruvian from Lima as a reference population. So while doing that, in Blacks, we will be able to reclassify our subgroup of Black and MVP into 3 categories. The first category is the Black with a high proportion of predominantly African ancestry at the locus. Then a second group, which is Black with mixed African and European ancestry at the locus. And the third group, a much smaller group, is a subset of Black with a predominantly European ancestry at the locus. So as we can see here, and as expected, 2/3 of our population have predominantly African ancestry at the locus. Now, if you perform the analysis stratified by each of these subgroups, what we see is that for the group of Black with predominantly African ancestry at the locus, we almost did not see any trend of association, despite about 11,000 cases in this group. When we look at the subgroup of Black with mixed African and European ancestry, we start to see a pretty substantial trend that looks like what we will see or expect in European, in white. And as you can see here, the number of cases is almost twice as less than the number of cases that we see in Black with predominantly African ancestry. In the subgroup of Black with European ancestry, unfortunately, we do not see any association, but certainly this can be explained by the fact that the number of cases is substantially low for this population. So in overall, what we're seeing is that there is no association between CAD and SNP at 9p21 among Black with African ancestry. So if we do the same analysis for Hispanic, to make it simple, we can reclassify Hispanic into 2 groups. Hispanic with non-African ancestry or low proportion of African ancestry, as well as Hispanic with predominantly African ancestry. And as expected, almost 3/4 of our Hispanic group have low or no African ancestry at the locus. So here I'm presenting the result of this subgroup only here compared to when we have our overall Hispanic plus external cohort combined. So as you can see with this locusome plot, for our overall Hispanic and external cohort combined, we're having a pretty good trending of association at the locus, but we're still not reaching genome-wide significance. But if you look at the subset of MVP Hispanic, we know a low proportion of African ancestry at the locus. We are seeing the almost exact same trend as we see earlier, and there is one SNP that even reached normal significance. And if you can see in the case control ratio here, we have again twice as less cases than what we have in the overall main sample of Hispanic. So what we say is that the association with CAD at the locus is stronger when we exclude Hispanic with predominantly African ancestry, confirming what we observed earlier, that the association at the locus is diluted by African ancestry or probably some African haplotype. So to be able to understand what could be the that specific haplotype pattern diluting the association with CAD, so the next step was to perform some haplotype inference and analyze the association with CAD for the haplotype in each ancestry group, and as well as in each of those ancestry subgroup based on a proportion of local ancestry estimate at the locus. So here we perform a haplotype inference using a subset of independent SNP that have an allelic frequency of over 5%. And we first selected a subset of SNP that reached enough wide significance in white. And for elder pruning to have our subset of independent SNP, we used black with predominantly African ancestry at the locus as a reference. So these together give us we started with 120 genotype SNP, just to make sure that we have a pretty accurate genotype calling here. And we ended up with 6 SNP that we use for our haplotype inference. So this table represents the subset of SNP that we use in the haplotype inference. So we have here the allelic frequency of those SNP in white, and here the allelic frequency of those SNP in black with predominantly African ancestry. So with these 6 SNP combined, we were able to have about 20 haplotypes that had the allelic frequency of over 10 to the -4. So this histogram plot represents the frequency of the most common haplotype combination that we have with this subset of SNP, here represented for white. So what we see is that we have 2 main haplotypes that represent over 85% of the haplotype frequency in all the haplotype combinations for white. And those 2 haplotypes are mainly based off of all risk allele of the 6 SNP for the first haplotype, AAC, ATT, or all the reference allele for these 6 SNP, GG, TT. So if you look at this haplotype frequency in Hispanic, we see a same trend with a lower frequency for these 2 haplotypes, but in overall, the pattern of haplotype frequency is similar to what we see in white. If you now look at the MVP, the overall MVP blacks, what we see is that these 2 haplotypes substantially drop in frequency and represent less than 20% of a haplotype frequency in all the haplotype combination, while we have another set of haplotypes that erase with a quite common frequency. And you can see that these haplotypes sometimes are pretty almost virtually inexistent in white. This probably reflects the diversity in haplotype diversity of African ancestry in general, or black in MVP. So now, if you look at this same haplotype distribution, comparing white with blacks based off of a proportion of ancestry, what we see, again here, the red represent the haplotype in white, the orange are the haplotype in blacks with predominantly European ancestry, the green are haplotype in black with mixed European and African ancestry, and the steel blue are the haplotype in black with predominantly African ancestry. So what we see, which is quite interesting, is that these 2 haplotypes that represent over 85% of the haplotype frequency in white are virtually inexistent in black with predominantly African ancestry. So the two haplotypes together have less than 1% of frequency, while we have a wide range of other haplotypes that erase with a quite distant frequency in black with African ancestry. So now, if you look at the association between this haplotype and CAD, so these two correlation plots represent the association between CAD and the haplotype. For each of the boxes represent white, blacks with European ancestry, blacks with mixed African and European ancestry, and black with predominantly African ancestry. And same thing for the second part. So in this first block, we use the most common haplotype in white as a reference and comparing this haplotype to all the other haplotypes. So what we see is that in white, when comparing this haplotype to other haplotypes, we see a pretty strong association for this and the second most common haplotype in white. Similar trend is observed for blacks and other subgroup. And we also see similar trend for black with predominantly African ancestry. However, this particular result is quite difficult to analyze or make any conclusion, since the frequency of those haplotypes are really, really rare in black with predominantly African ancestry. So if now we use the most common haplotype in blacks as a reference, so what we see is that this haplotype actually is also pretty rare in white. So it makes it difficult to make any conclusion here, even though we see a quite inverse association. But the interesting finding is that when we use this haplotype as a reference, none of the other haplotypes that are common in blacks show a significant association. So in overall, what we're seeing is that there are two haplotypes in white that are pretty frequent, and those two haplotypes really help to discriminate the risk of CAD at this locus, while those two haplotypes is almost inexistent in blacks, and particularly black with high proportion of African ancestry at the locus, probably explaining why it's getting really, really hard to be able to find the association with CAD in overall in that locus. So to summarize, to re-identify new CAD loci in white, black, Hispanic, and in our combined multi-ethnic meta-analysis, we show that with our trans-ethnic meta-analysis, we substantially improve the number of GY's local loci discovery when compared to ancestry-specific meta-analysis. And more importantly, we show that the haplotype discriminating the risk of CAD at 9p21 among white is virtually absent among blacks with a high proportion of African ancestry at the locus, probably explaining why it has been really hard to find the association with that specific locus, and also might contribute to why the progeny score is so not performing well in those populations. So with this, I would like to acknowledge all our collaborators and thanks to Team Assignments, my PI here at Stanford, and the MVP in general and the MVP investigator for the opportunity to work on this. And thank you all for your attention.
Thanks so much, Catherine. That was a great talk. We just have time probably for one quick question. Actually, I'm going to ask two of them together. So one of them was just like a technical question, which is, what did you use as the percent ancestry threshold when you were saying that somebody was sort of more African ancestry or more mixed? Was there like a particular threshold that you used?
Yes, so here we use 80%. So when an individual have over 80% of an ancestry, we use that as a predominantly African ancestry. Everyone between 20 to 80% were considered as mixed, and everyone over 80% of ancestry for white were considered as blacks with white ancestry at the locus.
Okay, all right, thank you for clarifying. And then one last question. Do these results suggest that the most power in analysis would come from pooling all participants and adjusting for ancestry with principal components rather than stratifying by the ancestry groups? Is that what you're saying? Because one thing I wondered about is you're talking about a case of genetic heterogeneity. Is that correct? Where
yes,
you can have different genetic architecture in different groups. So do you actually dilute some of the signal when you put them together or not?
So that's the point. I think what we see with, at least with Black and Hispanic, is that even though we have been over time adjusting in ancestry within population with global ancestry, we still see that there is a dilution of signal. So there might be some other type of way to really narrow down that association, but the sample adjustment is still not helping to understand what is going on.
Right. Okay. Thank you so much, Catherine. We need to move on to our last speaker of the day, who is Dr. Gary Gibbons, is the director of the National Heart, Lung, and Blood Institute at the National Institutes of Health, where he oversees the third largest institute at NIH. I think I'm going to let him do all the speaking about the important work that NHLBI is doing to promote diversity in human genetic studies. Welcome, Dr. Gibbons.
Okay, great. Thank you, Sarah and Dan, for this invitation and participation in the Super Symposium. My talk will be a little bit different from the perspective of as a director of the NHLBI and provide a sort of higher level view of some of the approaches we're taking to address the challenges relevant to this discussion. So as part of the NHLBI strategic vision, we have engaged our overall community in addressing our priorities. And again, I think relevant to this symposium, there are several that align with our strategic goals and objectives. And I'll touch on four elements of that. One is that perhaps our most sacrosanct mission at NIH is to nurture the next generation of scientific leaders. And it's critical, we believe, in order for science to continue to be innovative and advancing, is to be promoting inclusive excellence in our biomedical workforce. So I'll touch on that briefly. Similarly, as has been discussed throughout today and subsequently, we have recognized the importance of studying population differences and addressing health disparities as a high priority for our institute and NIH in reflection of some of the challenges in health of our country and around the world. A third sort of element is the clinical and implementation research that is critical, I believe, to have discovery science have public health impact. In that regard, we hope to foster inclusive participation in clinical and genomic research as a broad and sort of interwoven fabric of our science. And then finally, again, germane to, I believe, this symposium, we believe that all these things need to really come together if we're going to affect the promise of precision medicine, that diverse genomic resources enhance minority health and inform the health of all. And so really a key thematic element is the diversity and inclusion catalyzes scientific innovation. In that regard, we recognize that there are some challenges that we face as a broad biomedical community. And really it's a call, if you will, of collective responsibility that we address inclusion on this path to precision medicine with diversity in the NIH biomedical workforce. We believe that this is an area that needs a substantial amount of work, as you can see. The slide on the pie chart on the left showing the U.S. population and the diversity of our country. But on the right, you can see that the R01 pool is not particularly reflective of that diversity. And so it really, I believe, begins with the people. I know there's been a lot of emphasis on the DNA that we have and who it's derived from, but we also want to address the scientists as well. And we believe that those are interwoven. Indeed, there's data that supports. This is something that goes beyond political correctness, that inclusive excellence is a driver of innovation and scientific advances, that the process of ideation and coming up with new perspectives, new concepts, interactions between different disciplines, we believe is critical, essential to scientific advances, certainly in the 21st century. And there indeed is an evidence base for diversity in the workforce, that it leads to better problem solving, provokes deeper thinking, and broadens perspectives, better decision making, and drives research based on that lived experience that, again, could be particularly helpful in addressing health disparities. And so the data is actually pretty compelling about the evidence base for promoting inclusive excellence. In that regard, as this community thinks about this, I'm delighted about the diversity of the panel. I've noticed in your symposium, Sarah, and I believe that it was great to have Catherine lead in that we want to be sure that all the elements of the pipeline and trajectory of becoming a scientist and doing science is inclusive. And this audience recognizes that there are a variety of NIH programs that are designed to not only to nurture the next generation, but ensure, again, that it's an inclusive and diverse next generation of scientific leaders. There are just a few acronyms of programs. A couple I'll highlight, though, is one's toward the bottom there, the NIGMS Mosaic program. That's actually been signed on by practically all the institutes and centers in NIH that targets faculty development through a K99 R00 program, and a recently launched NIH First program, which is a faculty institutional recruitment program that promotes what we hope will be transformative cultural change at various academic centers around the country, again, toward promoting inclusive excellence and developing a diverse workforce. Clearly, we believe that brilliance and talent it comes in all sizes, shapes, and colors, and from around the world, and that's what makes us great, and that's what will be critical for an innovative scientific workforce of the future. Toward that end, just to give you a little vignette here, this first round of the NIH Mosaic came out, and want to give some a shout out to one of the investigators who is being supported by the NHLBI as part of that Mosaic program, and looking at various elements of sex hormones and chromosomes in Baxter oxidative stress. And so part of, we believe, promoting inclusive excellence is really to nurture the future of these new leaders. And we hope to do that on a global scale of inclusive excellence. And so I was also pleased to see the highlighting of H3 Africa and its process of promoting capacity building and training as an intrinsic part of doing the genomic science that's in the H3 Africa program that has been detailed earlier, has been very effective in bringing together a consortium of African scientists who are really driving genomic science on that continent. And it really raises a potential question that I'll put out as a what if. The African scientists created a Pan-African top-mid-like genome-phenome resource for the world, as well as something that would drive precision health, precision medicine on the continent, relevant to both communicable and non-communicable diseases that face that population. I put that out as a what if, a challenge, and an opportunity. So I'll pivot to just a few vignettes in the space of where there are health inequities and relevant to our portfolio to really start to address how in engaging the community in important issues of health inequities and racial disparities in health is to appreciate that multidimensional challenge and the importance and critical driving role of the interplay between genes and environment, and particularly the social determinants of health as part of defining a personalized risk and hopefully precision medicine. So it's really this integrated view that I think this symposium is hopefully fostering and hopefully will catalyze within this community. So as one element of that that's already been discussed that I won't belabor here is this challenge that we have of the skewed sort of pathway that we're on currently, where there's this large preponderance of sort of Eurocentric view of genomic research that is part of this field. And the consequences on the right-hand panel that have been addressed by earlier speakers about what that means in terms of assessing complex traits, many of which, again, have health disparity kind of consequences. And really, for me as a non-geneticist, really challenging this community to develop the technologies, tools, and resources, and particularly population resources, to really address this shortcoming of the field, given the compromising utility of asking and answering the compelling questions that need to be made. And so we really have to address this stark lack of racial ethnic diversity in genetic studies and clinical research in order to ensure that these elements have clinical utility for the populations that often are disproportionately affected by these disorders. Just as one example that this community knows very well, that when there is this skew and a lack of diversity in these genome-phenome resources, it could cause significant issues of misclassifications, of variants, because of the lack of appreciation of differences in allele frequencies, as well as implications about their potential biological significance or pathological significance. And so this has been documented and clearly needs to be addressed. In that regard, some of that was part of the motivation and intention that the NHLBI had in establishing the Transomics for Precision Medicine program, or TOPMed, in which, again, we thought it was critical to take an inclusive approach in developing this genomic phenome resource. Again, less about moral imperatives or political correctness, but more about the scientific integrity of our data sources, and that it should be more reflective of the diversity of the human family, and recognizing that many of the roots of that family indeed are part of the genomic variation that will inform biology and precision medicine relevant to the entire human family. So that was a critical element of intention in developing this resource, which now has over 150,000 whole genomes reflective of this ancestry distribution and a variety of phenotypes, obviously a bit skewed toward the NHLBI mission and portfolio, as shown here. And as a resource, something that we've also created in a cloud-based platform that we think can also promote democratization and access to anyone in the world capable of an internet access. In that regard, the embedding of TOPMed in our biodata catalyst in this cloud-based platform, we believe has provided some useful resources for the larger global community, and in many ways we hope has helped to democratize and promote access to data that maximizes discovery in that way. And many of you are familiar with the imputation server that's part of TOPMed. We heard from Gonzalo earlier that leverage this resource. And on the right, you can see how this has facilitated imputation in different populations. This one's the NHLBI's Jackson Heart study, and what we hope will do in promoting advances in genomic science by integrating these kind of resources, and then putting it in a platform where even if you don't have as big a cluster of computer nodes that Gonzalo used to have at Michigan, that again, even if you're hopefully in Accra, Ghana, or someplace, you can also leverage this kind of resource eventually. And so that's the intent and the goal. With that vignette, There, let me move to a couple of examples of health disparity conditions that might leverage these capabilities. Starting with asthma, which in many ways is clearly a health disparity condition that's particularly prevalent amongst African Americans, Latinos, particularly a problem amongst children, major causes of hospitalizations and indeed death, with major racial ethnic differences as noted here. But it's also almost paradigmatic of a disease in which there can be genetic susceptibility, but clearly an important environmental and gene-environment interaction component, whether that's where your neighborhood is and your exposures to air pollution, which again tends to be more problematic in Black and Brown, racially segregated communities, which known tend to have more toxin exposure, more air pollution, as well as environmental allergens and that interaction with the immune system. What's noted as these drivers is, again, racial differences. And on the left, you can see sort of a common pulmonary function test in which the reference panels are actually broken out by race ethnicity, in this case, I think for FEV1. And you can see these sort of patterns based on reference. And in many things in medicine, reference means studies they did in the 50s and 60s on white males, some of whom were medical students, to set up these curves and weren't particularly inclusive. Nevertheless, Esteban Burchard, a Latino investigator in TopMed, has been exploring this issue as cohorts down in Puerto Rico, that mixed population, obviously, and has been using a variety of techniques, mixture mapping, leveraging the whole genome sequencing that's been in TopMed, has been identifying some of the genetic determinants of asthma in these populations. And this is just one example of where it's used these techniques along with certain functional assays to identify a locus that is associated with this parameter of lung volume and lung function. And it does indeed seem to be at a locus that modulates the immune system, particularly interleukins, et cetera, and perhaps even agents that might relate to the therapies and the anti-inflammatory therapies that are useful for asthma. And so some of the utility of again, leveraging diverse cohorts using these technologies that may inform human health and disease. And I might also add, parenthetically, that passion that Esteban has shown, I think, reflects how important it is often to have an inclusive scientific workforce pursuing those questions based on their own lived experience and their passion as clinician scientists. Another important area of health disparity relates to women's health, and particularly women in reproductive age groups in which we have shockingly high burdens of maternal morbidity and mortality. It's particularly prevalent amongst African American and Latino communities, also American Indian, that are incredibly high. The United States has an embarrassingly high rate of maternal morbidity and mortality for such a rich and developed country. And you can see these racial ethnic disparities are particularly prominent and relevant to the NHLBI portfolio. Many of those pregnancy complications relate to both the heart and blood systems, whether it's hypertension, preeclampsia, peripartum cardiomyopathy, or blood disorders. And in the case of peripartum cardiomyopathy, where these women almost as part of a stress test of pregnancy, if you will, often become manifest that they have this dysfunction of the heart. Many of these women are African American, and it can be associated with the hypertension that can occur in pregnancy or preeclampsia. And there's been a hint of genetic predisposition in this regard, and where identifying those at risk could have important health and medical advantage. And a study done by Cricket Seidman and others has looked at doing sequencing on some of these individuals, and notably, it was found that many of these women actually do have mutations of the sarcomere, which are also seen in various familial forms of cardiomyopathy. In this case, with the Titin gene, again, a locus that is causal for a number of other cardiomyopathies. And so it is potentially the case that they may have mutations that in the context of the stress of pregnancy become manifest and may even presage a full-blown development of cardiomyopathy much later in life. The challenge, of course, is that what we know about sarcomere mutations is often limited by the, again, Eurocentric nature of a lot of those familial studies. And let me conclude with the last one, in which certainly these other elements of cardiovascular health really offer an opportunity to really understand how, again, this interplay of genes and environment behaviors, this multidimensional nature of how health is holistically defined, it involves a dynamic. And the left panel just shows you, I believe, a figure from Sake in New England Journal several years ago, in which he looked at increasing genetic risk profile and from low to moderate high in cohort studies from the NHLBI, but also how that appeared to be moderated or mitigated by the lifestyle profile of these same individuals. And again, indicating that a high risk potentially could be, by genetics, could be mitigated by taking a healthier lifestyle and reducing that risk. And clearly in the NHLBI portfolio, studies have been done with the DASH diet that was developed that showed that the diet rich in fruits and vegetables could lower blood pressure. And indeed, what we're learning is that it may do so by modulating the microbiome. And by modulating the microbiome, that also appears to modulate the metabolome. And so you can potentially see an interaction here in which by a transomic analysis, we might start to understand the mediator pathways that confer the effect of diet on genetic risk and how it's modified and what pathways may mediate that. That's hopefully the potential of a resource like TopMed. Similarly, this audience is quite familiar with the locus of PCSK9, and I think it's part of its discovery, I think, is notable in the sense that when Helen Hobbs pursued this and looking at sort of the tails of the distribution in the Dallas Heart Study, one of the, I think, benefits of it being so inclusive as a biracial cohort was that the African Americans, when she was able to look at that distribution, particularly the tails, it was obviously the African Americans that were particularly informative and gave the hints of those at the lower end and what variants were driving both very low cholesterol and very high cholesterol. And again, that aligned with some of the familial studies of both loss and gain of function at this PCSK9 locus. And as many are aware, that helped drive a drug discovery and drug development paradigm that had led to therapeutic agents targeting PCSK9 that were shown to lower cholesterol and eventually were shown to prevent heart attacks. And so sort of full circle in discovery science, from literally population science, bench science, to the clinical trials and now clinical practice. but underscores how important inclusive participation was in helping to drive that scientific discovery. Similarly, most recently, those sorts of studies were extended further in that the trial that I showed you, Reduced MI, was a study that looked at, if you will, all comers at high risk for having a heart attack, either had one before or had a lot of risk factors or both, that would suggest a benefit from aggressive lowering of their cholesterol with a PCSK9 inhibitor. And a subsequent analysis of these kinds of studies has applied genetic risk scores and shown that those who were at the highest genetic risk score amongst these trials appeared to have the greatest benefit of the cholesterol lowering. Again, aligned with what we're hoping precision medicine may bring, and where we move from a sort of one-size-fits-all to one that might be more tailored to that risk. And that's where, again, we're hopeful that advances in this community will refine our ability to treat and get the right drug to the right individual at the right time. The challenge, of course, is as in this study, 99% of the individuals that looked at this risk score and benefit of this drug were European. So I just wanted to quickly wrap up here about the scientific imperative of diversity and inclusive excellence in precision medicine, and really a call to the community of a collective challenge, a collective commitment, a collective responsibility to lean in to this collective effort to advance science in ways that enhances human health. That's indeed the mission of NIH. And we can only do that, in my view, if we're inclusive of all communities and that we generate science that has benefit to all communities, both in this country and around the globe. I thank you for your attention.
Thank you so much, Dr. Gibbons, for an outstanding presentation, highlighting many of the points that we're probably also going to touch on during the discussion. But there are two questions that were specifically related to TopMed. So the first question is, can researchers from outside of the US contribute genomes to TopMed? And can it be done before publication, during an embargo period?
Ooh, that's a tough one for the director as opposed to the staff leaders of it. So a few things. So TopMed, as you know, Sarah, what involved a process of really peer-reviewed prioritization of certain collections, in which the notion was, one, a commitment to share the data, B, obviously the NHLBI had an orientation to having robust phenotypes that go with that DNA to be submitted for whole genome sequencing and inclusion in TopMed, as well as, quite frankly, a prioritization around having particularly multidimensional elements to that submission. So it wasn't just about, again, the DNA alone, but we tended to prioritize data sets that had broad phenotypic spectrum, balance, as well as depth, because TopMed, again, is more than a DNA resource. It is meant to be transomic and also to have a number of different modalities, such that someone could just pursue understanding social determinants of health, as well as environmental influences in looking at that data set. And that was, as I say, through a peer review kind of process. TopMed did and does have, as with many NIH studies, some of the policies related to embargoes, et cetera, with regard to that contribution. Increasingly, now that all that's in dbGaP, we hope to continue to expand access to the TopMed resource, such that it's more open, accessible, and inclusive of anybody who wants to probe the data set under controlled access. And that's increasingly happening. So hopefully that
So,
addresses the question.
so thank you. Actually, and what you said about the importance of the phenotypes ties into the second question, which is, are there data related to social, clinical, and environmental risk factors that are available from the TopMed resource? Or is it all just sort of clinical phenotypes? Is there any information about environment?
So it's a great question. I think previous speaker talked about something like geospatial coding. And so to a certain extent, there is fairly, I'll say, granular data once you can get in by controlled access to the point where it would be possible to find out someone's zip code or census tract potentially, and the capability of linking that sort of data. Similarly, there are a lot of these cohorts, whether using personal sensor data or questionnaire data, have things like sleep and smoking behavior, exercise activity, et cetera, et cetera. So it's a pretty broad set of data types, data elements. Clearly, literally people have been imaged from head to toe in a sense, brain MRIs, chest CTs, abdominal CTs. So there's a tremendous array of different types of data to ask a lot of different kinds of questions.
That's great. It's an amazing resource. And at this point, to my knowledge, it's the largest whole genome sequence, high coverage whole genome sequence data set in the US with the most diverse set of participants or subjects. So anyhow, let me now go ahead and invite all the speakers to join us. So please, everybody turn on your video, turn on your mics. And if you, there we go, perfect. And unfortunately, Dr. Abecassis sends his regrets that he can't join us, but we're missing Dr. Hanchard, and there he is. Fantastic. Okay, so we're going to talk about some of the general themes that are, and I should say, even though we're running over time, we still have 30 minutes. We're doing fine. So we'll talk for about 30 minutes. So a couple of things that came out of this discussion today. Clearly, we need to do better. We as a community, a scientific community, need to do better to include more diverse individuals in human genetics research, or we could say biomedical research more broadly. And then secondly, we heard a number of presentations about the importance. What are you going to learn if you include diverse populations? And it's of relevance not just to those populations, but to everyone, right? Because a lot of this information benefits everyone. So I guess the question now is really, how are we going to achieve this? What are some of the boundaries or challenges, I guess, of including more diversity? And actually, Dr. Gibbons, you touched on this point, and somebody asked a question, see if I can find it, because they pointed out a lot of the discussion is about, oh, we need more diversity of subjects and so on. And said, without representatives, I think they meant of people of diverse ancestry playing a role. Do we risk exasperating disparities even further? Isn't the training of individuals from diverse backgrounds just as important? And you made it clear, Dr. Gibbons, that there's a lot of effort at NIH to increase funding. But what are some of the other things that need to be done, whether that's education at the earliest level, or what can we do at our universities to try to have a more diverse workplace? And I'll let anybody jump in here.
Well, hey, I'll sort of start. I think, part of the reason I sort of start off with Esteban's study is that, I've known him literally since he was a medical student. I was at the faculty at Stanford, that what he is studying, I think, is derived from his lived experience, as a clinician scientist as well, and his passion. So it's not surprising that he would take on, what's driving asthma in Puerto Rican kids, and bring his expertise to that question. And so for him to do the outreach, to go to Puerto Rico, to make the links and to make it happen, that's something that clearly I think reflects his drive and passion and talent. I think that's one of the side products, if you will, of being inclusive, because that perspective, that lived experience, I think, adds to the mix of all the top med investigators. And in that case, I think has helped elucidate and leverage the resource and made it a more effective one. So that's where I think the people are also important. And then I think this was touched on, I think, by Neil a bit, is that when you're reaching out to these populations, for the most part, these communities do want to participate, but they want to be asked. And so sometimes there's a little bit of victim blame, and somehow there's some resistance or hesitancy to participate. I think, at least it's been my experience in recruiting communities of color, that once it's explained why we're doing what we're doing, that the questions that we're asking are relevant to their families, their neighbors, their community, and it might address issues, problems, challenges that they're very familiar with in their communities. Often the participation is quite good. And so then it's a matter of sort of our responsibility to ensure the data actually gets back and has results and impact on that community. If there's that virtuous cycle in the same way it was talked about of a learning health system, but we see that at a community level with these communities, I think that can broaden that diversity of our studies. I'll stop there.
Yeah, that's awesome. Anybody else want to jump in about your own personal experiences?
I'd be happy to.
Okay, Marilyn.
So one of the roles that I have at Penn is co-chair of our Equity, Diversity, and Inclusion Committee in our Genomics and Computational Biology graduate program. This is a committee that's about half faculty and half students. And one of our goals is exactly that, how to get more of the underrepresented communities into the pipeline of computational biology and genomics. And there's a lot of efforts ongoing. Dr. Gibbons presented several, the FIRST program at NIH and others at the faculty level. And I was so happy to see the slide that started with high school, because I think that's where we have to start to reach individuals, many individuals who grow up, in particular in low-income communities and, without academic kind of hierarchies in their family. And I'm one of those. I'm a first-generation college student in my family. I didn't even know graduate school was an option for me. I didn't know about genomics. And so, even as a white person, I'm then, so, for my peers growing up, who were Black, had they weren't even sure they were going to make it through high school, much less go to college. And so I think the more that we can reach high school students and make them aware of opportunities, and then influx the resources to actually, provide opportunities and to mentor them through, it's not enough to tell high school students, look at all this cool stuff that you can't do. It's how do we get them through high school and into undergrad, and then from undergrad into medical school or graduate school, or whatever path is the right path for them. But I think we need to start early, because we keep talking about recruiting more faculty, from diverse communities, and there just aren't enough people because we have failed them earlier on in their education. And so they have fallen off of that path much earlier. And so I'm really passionate about reaching them sooner.
Yeah, you make some excellent points, including what do we call diversity, right? So it can include first generation, college, people going to college for the first time. It can include people with disabilities. It can include even women. And unfortunately, in my field, there aren't as many of us at the advanced levels. But I'm just wondering, I don't know, Catherine, if you want to add anything about your own sort of pathway or how you got to be where you are today.
Can you hear me?
Yes.
Yeah, I think for me as not a represented person in science, it has always been a challenge, and I think a lot of us face not only the challenge of representation, but also the fact that we always have to prove that we are the best out of all other people around us that are much more represented. So I guess at our level, and with all the initiatives that we started out there, like Black in Cardio, Black in X in general, the objective is to say, hey, especially for trainee and people like us in the transition phase, we want to be able to say, hey, we are here. And I think one of the biggest frustration is that when it go from trainee to faculty, there is these things about all at all academic level where we want the best of all candidates, but then we have to prove ourselves that we are the best. But also, I think it's not acknowledged enough that in the pool of people where we have hundreds of white and 3 blacks, it's going to be hard to have the 3 blacks that are the best out of the 100 of white. And this part has not been well acknowledged right now. And we hope that at least at the leadership level, they'll be able to understand that the representation that we're talking about and the representation in science start with looking at the pool of those individuals and select them based off of their capacity without comparing them with the entire pool.
Right. And we have to be like proactive, right? Find the best people. They're there. Let's find people and give them the support that they need to succeed, because that's the other thing that's really important. Actually, Neil, because I wanted to touch, we talked a little bit about, Dr. Gibbons mentioned people's willingness to participate. And I'm wondering if you can comment on that from the H3 Africa perspective. I know from my own research that generally people were very willing to participate. We have to be careful not to be coercive, right? But it depends on the community. And tomorrow we'll start talking about maybe in American Indians, it's very different. But what was it like from your perspective?
Yeah, I mean, I can give a few perspectives here. One of them coming from, so I'm also a part of the Diversity and Inclusion Task Force of the American Society of Human Genetics, where this has obviously been a forefront. So there are efforts such as the Human Genetic Scholars Initiative, which is designed to take individuals from who are sort of promising postdocs and early career scientists, and be able to provide for them the mentorship and the background that they need in order to sort of have the sort of insights to things that they need to have, but also to pursue some of their efforts to sort of lift as you rise, so to speak. And so that's sort of one example, and also sort of collaborating with NHGRI to sort of push the idea that, we want to have a breadth of researchers in those positions. I think for the recruitment side, there's sort of 2 aspects to it, which you kind of alluded to. As you said, it depends on the community. I've generally found, recruiting in Jamaica and Botswana and in Uganda, that there's a lot more willingness to participate. There's a lot more of this community effort. There's a lot of resistance to the idea of sort of colonial research, right, where the people swoop in and there's not a sort of quid pro quo, for lack of a better term, where there's some sort of development that occurs on the other side. And often that tends to be the biggest sort of roadblock to things. People want to participate. Right. The communities are happy to participate. The researchers are very wary because they, just don't want to see people sort of making their careers and they sort of get left behind, or, the countries get left behind and don't have the ability to continue to develop. So I think that has been something in H3 Africa that's been a source of a great deal of contention, because obviously these are, a lot of the funding that started this is public funds and it's for the public good, but how to do it in an equitable fashion so that the main purpose of the grant and the consortium is still met is something that, becomes a debate about how you can create that equity. I think in the US, it's very different. This is a conversation we've had as part of the diversity task force. Is there is this long history, and one of the questions alluded to this, of marginalization, stigmatization, and in some cases just like ethical negligence, which has to be overcome. And one of the things that has come out from those conversations is that there needs to be a truth telling, right? So people come clean with, the facts of the past. They are the facts. But then also sort of give a hope that, we've learned from those mistakes, that there's a new direction. We've learned a lot more about how people view ancestry and genetics, and that going forward, you sort of have to change the narrative. And I know you have speakers who are coming tomorrow to talk about changing the narrative and how we speak about things. But I think that's what people are viewing as an important part of that. And then engaging those communities. The society is in the midst of trying to create a document that really speaks to that issue about how to engage communities properly. So just like Dr. Gibbons talked about, actually approaching them, asking them if they'd like to participate, engaging them in a longitudinal way, not just get in there, get some samples and move on. But creating a community engagement of to and fro, allowing them to be able to say no if they don't want to participate, and being just transparent and open and bringing them to a real seat at the table, not just a kind of ethereal one.
Right, and it seems like community engagement is really key, right? That people from the communities be the ones leading, the research, or at least asking the questions and so on, and that they have to be included at every level, right? Starting at the very beginning when these research is being constructed. So Neil, you touched on something, one of your comments made me think about the question of benefit sharing, okay? And then that got me thinking about something that Dr. Gibbons mentioned, which is, what is it, the PCSK9 gene, right? And wasn't that the one then that it was commercialized? There's a therapeutic that was actually developed by Regeneron. Unfortunately, we don't have Dr. Abacasas on right now. And that sort of raises this question then about commercialization of information that we might get from these diverse populations, and how are they going to benefit? How are we going to do and what is benefit sharing? Are there different types of ways that people can benefit? I open this up to anybody.
Well, yeah, no, I think it's a great point, and it really extends off of Neil's point about the community. At least from where we work, we see this as a partnership, and so if you're partners, then there should be mutual respect, mutual benefit, and so in that sense, the notion of, as you point out, sort of helicoptering in, getting some DNA, going back and writing your papers, that's not going to be a paradigm that's going to engender both the trust, the buy-in, and the participation. And that's why I thought his point about the longitudinal element is key. That's where I would hope that as we study all these DNA variants, we're still tied as a clinician scientist to the health of those communities. It does no good if we took their DNA and we return back a polygenic risk score that they can't use. So that's the part where engaging, as you say, from the beginning, having these communities at the table when we're designing these things, designing these studies, analyzing the data, interpreting the data, and then translating it into the advance, whether it's a new therapeutic or how to make patients or achieve better outcomes. So that's where I sort of threw out that what if of H3 Africa continuing to evolve to the point where it's part of a learning set of health systems within and health ministries across the continent as well, and in a way that's very useful. And in that way, I would also agree that it may be sort of unidimensional, again, as a non-geneticist, to think of it only from a DNA standpoint, because the community thinks of these things holistically. And I think we need to do as well. And in fact, that community wisdom, I think, is good scientific wisdom. Those are the best models that we really need to have an impact. I'll stop there.
Anybody else want to comment on this, or ways we could do better at benefit sharing?
Yeah, I mean, I think that the benefit sharing is a big deal for the communities, yes, but also for the sort of researchers who are involved in it. And there's a lot of push and pull about how to accomplish that in an equitable fashion. And I don't think the answer is very clear to this point in time. But I'm so keen to hear sort of tangible ways in which we can do this. And, I think the community engagement obviously is central to how you get these things done. But how do we ensure that it actually gets done? So, one of the conversations we've been having in this task force is, it takes a lot of time to, contract with communities up front and make decisions about what you need to do. That time isn't always allocated for in a grant. You don't really get money to do that kind of thing.
Yeah.
Or it doesn't necessarily reflect when you go up for tenure, or any of these kinds of things that are outcomes on the other end. Journals don't necessarily ask you to talk about what's your community engagement. And so some of the conversation we've been having is how can we sort of build in systems or sort of advocate for systems that will sort of insist or make it a very natural process that we can have more benefit sharing. And that's a tough question.
Well, I think, and tomorrow, I think, that's also going to come up as well when we talk about benefit sharing. But one thing that came up, somebody asked a question, they addressed it to Dr. Gibbons, but it's really not fair to make you be responsible for this because it's something I've thought about as well, which is that, okay, so we've shown that larger sample sizes matter, particularly when doing a GWAS if you have lots of variants of small effect. And there may possibly be a different genetic architecture in different populations. So we have to include these more diverse groups. But it's challenging for the reasons we just discussed. So we may not have as big numbers, right, as you have in European data sets, which are now in the hundreds of thousands and going into the millions. And one of the things that this person mentions, and it also is a concern of mine, is that we get dinged when we apply for an NIH grant often if we either have smaller sample sizes and they say, nope, you're not going to have the power, and so we get actually penalized for proposing to include diverse populations because they're not as big. And then likewise, when you go to publish in the journal and then the reviewer says your sample size is too small, or there's genetic heterogeneity, I don't trust this, so how can we overcome these issues?
Can I go first? Having sat on a lot of NIH study sections and reviewed papers for, I don't even count the journals anymore. So when I'm at study section, I am the wet blanket that I think everybody else gets annoyed when people start saying the sample size is too small. I'm like, if we don't allow them to do the study, it's never going to get any bigger. And so I think what we need to do is educate the peer reviewers and then find enough people who are willing to stand firm in the importance of the diversity of the populations, even if they're smaller. If we don't start now, they're never going to get bigger. And so we need someone in the room in every study section saying, that's not valid anymore. We need to study this population. And same thing with manuscripts. I used to be one of those people who, the European study was, figure 1 and the African analysis is in the supplement because it's not significant, and the reviewer made me hide it. And now I put them in a panel together. So at the very least, they have to make me split the figure. But I think it's educating the reviewer, having, at least at study section, somebody just has to be in the room and shut it down every time.
Or maybe give explicit instructions to the reviewers. Don't, mark people down because they're including diversity. We should there's information, as several of you shown, Catherine and Neil, and, you've shown that even with smaller, you may not have the biggest sample size, but given the diversity in Africa, for example, the genetic and phenotypic diversity, it may be more informative with smaller numbers, right? And then, or including diverse trans ancestry. Oh yes, Catherine, please. You're up.
I just want to continue highlighting what Maureen just said, because we literally had that exact same thing where we have been working in this study where we're almost the only people that try to highlight the tinier sample from other ancestry group. And you went through several reviews because one reviewer said, I don't trust these, the sample size is too small, you should remove these results and put in the supplementary. I think this was literally what was said, that because the sample size is small, we should put that in the supplementary. I got frustrated because, again, same reaction. Why is that? Because of the sample size, people have to not show this, not publish this, or have to find the very lowest impact journal to publish. Not that I'm undermining some impact journal, but the point is that the more representation we have, even in a tiny sample, the better we will get. And there is a substantial gain in publishing in showing data from smaller samples, because this is the only way we will be able to show the subset of data that we have to the world and give the possibility of combining those samples from everywhere. And the perfect example is the UK Biobank. So very few people know that UK Biobank have 2% of non-white. So there is literally 6,000 Blacks and African and Afro-Caribbean and 8,000 South Asian in the UK Biobank. But our study that include the UK Biobank systematically rule out those subset of individuals. And when you try to use that, this is not necessary. So, but how would we improve things? We are not able to show the minimum that we have. And thank you so much for bringing this, because I think either in the paper, at the published paper level, or at the grant level, I think something has to be done to have these coming out.
Yeah, I totally agree. Any other, final thoughts about that, or any other topic you want to talk about?
My pet peeve has often been, we tend to look at sort of how grants are allocated and, the proportions there, but there's always the gatekeeper issue that, tends to get maybe less press, but, it sort of perpetuates some of the same problems. Because often the gatekeepers and the people who sit on the study section are actually coming from the same pool, so you sort of perpetuate things to some extent. And the same thing happens, I think, at the level of sort of reviewers. And so I think it's incumbent upon editors and, higher ups to ensure that these are things that people are aware of and looking for. People have talked about whether we need to have like a equity statement in these things, or you need to have a community engagement statement, or, now I guess you get dinged for not having enough diversity. So that's a start, a step in the right direction. But we need to keep moving in that direction in order to get it done. And I think it's going to be, as it comes, the edicts that come from on high, unfortunately.
Okay, speaking coming from on high, that Dr. Gibbons.
Right. I am so high.
And that's the part where, if you noticed, I said our mission comes from a circle of partners, that it's collective. So I hear your point that obviously the NIH has an imprimatur and a bully pulpit and some power of the purse, but to really be owned. It really needs to be owned by the entire community, and not just something that we wag our fingers at. And so at the end of the day, it's on all of us, and it's really extending that notion of inclusion, in which at every level, not just the participants, not just the DNA donors, but in fact, every level of our science. How inclusive are we? Because as you're pointing out, the power is around all those tables, whether it's study section or the editorial board, and what have you. And we, as Sarah pointed out, at the leadership levels, these things tend to be less diverse and inclusive. And so that's also part of what we have to work on as a collective, where those decisions make, who's in the room, and what diversity of perspectives are in that room, to make the pitch that I'm so glad Marilyn made, that of these things. I also hope that, and this is where it goes beyond the on high. I'm delighted about this panel. I hope you're developing your own networks, your own consortia, that at some level, it also probably needs to be grassroots, where you find folks with shared interests and you develop your, African diaspora consortium, or whatever else you want to call it, or Pan-African consortia, and then challenge us to fund it. So
you heard it, guys, directly from the director of NHLBI. So don't forget that. You guys, we could just go on and on forever. This has just been an amazing discussion, but we got to stop. We're almost running out of time. But I want to thank you all so much for your excellent presentations, excellent discussion. Remind people that there is going to be a recording, and I hope people spread the word about this, because I think people are going to learn a lot from that discussion. And we're just going to end with some concluding remarks from Dr. Dan Rader, chair of the Department of Genetics. So Dan, we're going to pass it on over to you now.
Thank you very much, Sarah. Can you hear me?
We can, yep.
Great, great. So what a fabulous day one of this symposium. I have to say I've gotten a couple texts from students in my lab already saying how much they learned from today. So I really want to thank all the speakers for really terrific presentations that were highly complementary, I would say even synergistic in nature. And this panel discussion at the end was also really, really fabulous. I just wanted to make a few quick points and then we'll close for today. Certainly, I think you can tell Penn Genetics is highly committed to the broad inclusion of all ancestral groups in genomics research. We also have a number of investigators focusing on developing computational techniques specifically for the analysis of these diverse genomic studies. And we also intend to and are leading implementation of genomic information into clinical practice in ways that will advance precision and genomic medicine for all. And then finally, I'd like to echo Dr. Gibbons' comments and some of the discussion in the panel about the importance of diversity among the biomedical science workforce. Penn will be submitting an application to the NIH First Award that Dr. Gibbons mentioned. I'm very excited that genetics and computational genomics will be one of our major themes and areas for recruitment of underrepresented faculty as part of this application and in general, even if we don't get funded. Certainly, I'll say, anyone attending the symposium who's entering the job market, please reach out. And as mentioned at the panel discussion, we are working very hard to increase the diversity of our training pipeline as well. Finally, I wanted to mention that genetic counseling is a profession that is not very diverse at all. And we have one of the largest master's programs in genetic counseling at Penn in the country. And increasing diversity of the genetic counseling workforce is a critical goal of this program. Finally, and if I can, I'll try to bring up a slide here. Let me see if I can do that. I may not be able to do it, so I'm sorry.
I know. I was telling everybody it gets kind of tricky, but what you do is share it and then touch. You have to make sure you touch the slide presentation first.
Share screen and then touch the slide presentation. Okay. Well, that.
Dan, I can bring it up. I can bring it up actually. Just let me.
Here we go. Is that it?
Got it. Can't see
great.
it. We can't see it though. Try to touch on it one more time.
Okay, I'm sorry about that, but it's a very important slide. How about that? No? No? Okay. In any case, what I was trying to show is a slide of pictures of 4 individuals who really made this possible. Dr. Dorothy Hammond, Amina Alamin, Augustine Benjamin, and Kimberly Runyon, the folks behind the scenes who really did all the work to make this happen. So I really want to thank them very sincerely for all that they've done. So I'll close there and invite everyone to join us again at noon Eastern Time, US, tomorrow for the second half of this symposium. Thank you so much for attending today.